Okay, so I'll be talking about the field trials, and it'll be real quick. Just give you an idea of what we've planted, and we've gone through some of this before. First, just to reiterate that we've planted in Oklahoma, Tennessee, and Texas, and Oklahoma and Tennessee will report right after me. And we have 3 different trials, the genetic study, the cultivar trial, and a GWAS study. And the details will be discussed a little further once we get this done. But here in the first trial, when we first started planting, actually, we started planting in December of 21, but everybody else in 22, I believe, in Bixby and Crossville and Somerville in Texas. And those are the number of plants that we planted. So we have a complete set. And as we propagated, we did as many as we could in every other site. So we really have a really good collection of all the progenies and everything at all 3 sites. And so what we planted, we have, depending on the site, 650 to 850 unique individuals. And we looked at, we were interested in resistance to RRD as well as black spot. So the RRD resistance and the crosses we have there, the resistance is coming from a source from the Carolina rugosa family, and which is different from the previous source, which was from the Synstyles, so which was Churiana source. And so the source we have now is Morden Blush and Morden Fireglow. We have a whole range of crosses with that. And then for black spot, we're looking at partial resistance with resistance from Miracle on the Hudson. And then dominant from Rambling Red and Baby Love. And a lot of this work is being done from a race-specific point of view with the people in Minnesota and Wisconsin. And they will report on that. Let's see, next. If you look at the cultivar evaluation trials, again, planted in 2022 and 23. And just in Texas, I'll talk about, we put about 300 or 729 plants. We have 194 unique genotypes. And this is, the diversity here is really quite wide. I planned it that way. This is also planted in Oklahoma and Tennessee, and they'll report on their numbers. Again, 194 unique rose genotypes, broad range of diversity from all the way from Chinensis types to rugosa types to everything in between. And so a lot of shrubs and floribundas and you name it, we probably have it. So, and that's the whole idea. A lot of these were selected because they showed some hint of resistance in the previous trial, and then we added things to that. So it's just really very good stuff. Again, we look at the GWAS trial. Now, the GWAS was something we added after because in the original proposal it was not there. But talking, Oscar and I talked a lot about this, and it's an approach to try to identify new sources of resistance to various things. Of course, our emphasis is rose rosette and black spot. And so in Texas, this past year, we planted almost 900 plants and 497 unique accessions. Most of them have at least 2 replications, or some that one died, so we only had 1. And this is also planted in Tennessee. So in Tennessee, they'll do the rose rosette resistance screening. We'll do the black spot, cercospora, and a whole range of other traits. And again, broad range of diversity. The other thing here is that since when we put the GWAS in, I made sure there's a lot of overlap with the cultivar trial, which is right next to it, basically. So we have 78 genotypes in common. And so we'll be phenotyping the same years. And so we can combine those trials to do an even better GWAS and increase the numbers to 610 unique genotypes. Gives a lot more power in the search for new genes for various resistances and other traits. And I just want to point out that we got rooted liners and bare root plants from a wide range of people. And I appreciate all the help we got on the propagation of these materials. And again, to thank everybody who has supported the project over the years, and as we continue on to get this work done. And with that, if you have any questions, more than happy to answer them, but probably need to be on our way.